Welcome to the European College of Neuropsychopharmacology podcast, where we interview the most interesting people in translational neuroscience.
Hi everybody, here is another podcast of the ECMP. My name is Ilse Graat, I'm a member of the Early Career Advisory Panel, and sitting next to me is Anna Beyeler. Anna, thank you very much for being here.
Thank you for having me.
Yes. Anna Beyeler completed her PhD on spinal cord circuits in 2010 at the University of Bordeaux. Subsequently, she embarked on her research journey as a postdoctoral fellow at the Picower Institute for Learning and Memory at MIT. During this period, she focused on uncovering the circuitry and synaptic mechanisms that underlie emotions in the amygdala, particularly their role in the formation and retrieval of both positive and negative associations. After 5 years, she established her current research laboratory at the Neurocenter Magendy. Within this lab, her team delves into the understanding how circuits within the insular cortex contribute to emotional valence and anxiety, while also investigating how these circuits are altered in preclinical models of psychiatric disorders. In 2020, Dr. Beyeler achieved the status of tenured principal investigator at the French Institute of Health. She has also been the recipient of the INSERM Avenger Fund and holds memberships in prestigious organizations, including being an associate member of the American College of Neuropsychopharmacology. She was part of the scientific program committee of the 35th ECMP Congress in 2022. Anna is also an ambassador of the ALBA Network and a scholar of the FENS Network of Excellence, a network of junior and mid-career neuroscientists joining effort to shape the future of neuroscience by putting young researchers in the driver's seat. So Anna, in this podcast we will talk about the struggles of building a career in academia and of course also about your own scientific career. So to start, what attracted you to neuroscience in the first place?
Thank you for your introduction. I think it's a very difficult question because it's not like one detail that triggered my interest for neuroscience. I think it was really first my interest for biology and nature. I always wanted to understand, I grew up on the countryside and seeing all these insects and what was controlling their behavior. And so I was very curious about the world surrounding me. And the more I was studying, the more I realized that the brain might be the most fascinating organ to decipher. And so that's why I decided to study neuroscience. But I hesitated for a long time between genetics and neuroscience. And I ended up doing neuroscience because I thought that in the brains there are also genes, so I could maybe use genetics and study genetics within the brain, which is a little bit the case, and we're getting to do this whole transcriptomic analysis in specific circuits.
Cool. And you did a PhD on spinal cord circuits, and how did that happen? How did you get into this topic?
Well, I think what really got me into this PhD project was the neural circuits. I was fascinated by neural circuits. I had read a lot of psychology and thought that maybe there could be neural circuits underlying or human behaviors. And this was still in the level of hypotheses when I was a student. And I really wanted to study that, but there was not really any lab doing that with these complex behaviors. So I decided to go on the spinal cord, which is a more defined neural circuit. And the other aspect is that the lab I joined was using electrophysiology to study these circuits, and that was really a technique that I wanted to learn. And I was very lucky to be trained by my PhD mentor in the lab I joined for my PhD.
So that was a really good start of your career in academia.
Yeah, no, definitely. I mean, I really learned a lot during that time, and it was a very biology project. And I was working in the spinal cord, but in a other animal model, which is really far from preclinical. I was working in Xenopus, which is a frog, and I was studying how the spinal cord circuits were evolving over the metamorphosis of the frog from a tadpole to an adult four-limb swimming animal. So it really had this dimension of biology into neuroscience. I feel now what I'm doing is closer to maybe psychology and translational research than what I was doing there. And I mean, both are fascinating, and I really sometimes miss it. And I have friends that work in the not only spinal cord, but in frogs, and I'm always happy to see their papers coming out.
Yeah, must have been really fascinating. Like,
yeah, it's very different, and in this lab it was very diverse. There was a colleague working on the aplysia, which is sea slug, and we were actually once a year going on the Bassin d'Arcachon, which is it's on the coast, close to Bordeaux, and we would go fish these sea slugs for the experiment over the year. So yeah, it's very biology oriented start of my training.
yes, and also very diverse set of things you have to do then, like from the lab into nature.
Yeah, no.
Like,
So I didn't have field work specifically. I was just helping fishing the sea slugs for my colleagues.
Finding the sea slug. Yes, yeah. And then, of course, your interest shifted to mechanisms that underlie emotions.
So I guess the interest was always there, but there was no really, around me, especially in Bordeaux, there was no labs that were studying that. That is also why I thought it would be interesting to study circuits, control locomotion and simpler behavior to really gain knowledge, also technical knowledge. But the interest was always there. Actually, my very first internship was in a lab that studied the impact of a polymorphism of the pro-BDNF, which is a growth factor, in different strains of mice. And I did a lot of behavior and a lot of behavior quantification and a little bit of neural activity analysis with the CFOS. But that was my first internship. So the interest was already there.
Yes,
but then I didn't find a lab which was studying emotions with like a very general term and using circuits and electrophysiology. So I thought I would do it step by step.
Yes, yeah, and eventually you...
Yeah, exactly. So then I actually did a first short postdoc because the PhD in France is only 3 years. And then I completed my thesis and published 2 papers. And then I went to a first postdoc in Bordeaux, where I also studied neural circuits, but really microcircuits in the hippocampus that underlie gamma oscillations and theta oscillations, which was also like in the bigger scheme of one day applying this knowledge and technologies to understand the neural circuits of emotion and now specifically anxiety, which is the main interest of my lab.
Yeah, yeah. And you did a postdoc in France and in the United States?
Yeah, so I did this first postdoc of a little bit less than 2 years in France. And then altogether with my PhD, it was 5 years of training. And then I felt I was competitive enough to go abroad in the US and, apply to big labs because there the PhD is at least 5 years. Like 4 years is really short for a PhD in the US. So generally it's 5 years. So I was feeling that right after my PhD, I was not trained enough to start a postdoc in the US. So
yes, yeah.
After this first small postdoc, I felt I was ready,
confident, and
well, confident might not be the word.
Like you had all the experience, tools.
I thought it was worth applying.
Yeah. And what was the difference for you doing a postdoc in France and the United States?
Well, it's a very different environment, and I have to say also the U.S. You cannot generalize the U.S. as just one uniform place. There's a lot of diversity there too, and across university. And I joined MIT, which is really one of the top university with a lot of funding, a lot of space, a lot of support, amazing people from all around the world, and it was an incredible experience. I would say a bit richer than what I was living in Bordeaux, but I think in the US there are like equivalent places than where I did my first postdoc. And I have to say also that in Bordeaux there has been a huge change of the neuroscience community over the last 10 years. And now, well, maybe we're not MIT level, but there's really a huge growth of the neuroscience community. And also, which I think is very important, is a reunion of all the neuroscientists of Bordeaux in one campus. So now it's like 600 researchers, postdoc, PhDs, technicians, teachers, all together in the same location. So when there's a scientific event, you meet a lot of people. There's a lot of collaborations. There's grants to foster these collaborations. So yeah, I really like both experience, but although it's true, MIT is a special place.
Yes, yeah. What's your best memory of your time at MIT?
It's not like one single event,
No.
but it would be working in teams. I think this is something I didn't really had before, like during my PhD and first postdoc, where, you had your project, so you had your mentor you were working with. But at MIT, we really were like 2-3 PhD/postdocs working together on one question. And I think that was really amazing experience. And this is what I'm trying to cultivate in my lab, trying to bring people together to work together, that they have this experience. And, science is hard sometimes, and, you have failures. So you have some support in these moments. And when something works, and I mean, it's maybe amazing for your project, but if you go back to your family and friends and explain it, they will probably not understand. But if you have colleagues who have been contributing to that project too, they will be very happy with you as well. So
Yes,
I think, working daily with colleagues, like doing experiments together, like in parallel, like also this feeling of being more efficient because you can parallelize experiments. This was, I guess, these people are my friends forever now.
Yes, very, very important.
Yeah.
Also to learn from each other, but also, just be connected. And And your recent work gravitates on, like you said before, on anxiety, also on stress. Yeah, so what research or what paper are you most proud of?
Well,
maybe
that's
also,
hard.
again, difficult to find one thing. Or
Well, I guess for now, I'm the most proud of the paper that was just published a few weeks ago. It's the first physiology paper of the lab, like with a functional study of a neural circuit and that controls anxiety-related behaviors. So it's the neurons, specifically neurons from the insular cortex that target the basolateral amygdala and that have anxiogenic properties. And we really dissected how they encode information, anxiogenic environments, and stimuli of positive or negative valence, and how these activities are linked. And I think really this is the paper I'm the most proud of today. Although I was very happy, or I was very proud of what I did during my postdoc in the US as well. I really remember reading papers about the contribution of the amygdala in fear, and being a student learning about these papers, and always thinking about how is it responding to positive valence? Like, does it contribute in any way to encode information of positive valence? And at that time, actually a few years after, like during my PhD, I read the paper from Kay Tai, who became my postdoc mentor after, where she did like first experiments tackling this question. And I really wanted to push that further and really dissect neural circuits, because she found that indeed the amygdala also contributes to positive valence. So the question was like, but how is that possible? Like the same brain region doing everything, like both positive and negative. And one hypothesis was that different circuits, so depending on their projection, the neurons would encode the information differently. And this is basically what I found in the amygdala for the first time. And it was very exciting. And it was a team effort, so we were using different techniques with other people, and so that I'm also still proud of that work, and to see that it has an impact on the newer generation, that now when they study the amygdala, they don't think it's only the fear center. They know that it contributes to other behaviors as well. So that's
very important. Yeah.
So I don't know the impact that the paper that just got published from my lab will have, but it has a big impact for us and for the people that did the work. And the lead author just got a position now in academia. She's actually the first postdoc that will start her own lab after spending 4 years in the lab.
Amazing.
So I'm happy of that. But I'm also as proud of the other postdocs. So one is doing another postdoc at the Max Planck Institute in Jupiter, and 3 others are in industry. Actually, my first postdoc started a company to support research, and so I'm proud of them, I would say.
No, but very good that you mentioned this, because of course you also wrote a perspective on existing and emergent scientific career models in neuroscience. And you also emphasize that it's also important to look at other careers outside of academia.
No, and recognize these all careers in neuroscience. I think they're all as valuables and we need them all. And I really support people regardless of their choice, right? My job as a mentor is to guide them on their own path. And
how do you do that?
Well, it's a lot of talking and it's actually anticipating a lot. So from almost the very beginning of their postdoc or even PhD, like taking the time, like at least once a month to talk about future.
Yes.
I think this is what helps the most, is like projecting yourself in the future and where do you see yourself? What would you like to do? What do you like to do in your daily work? And how can that turn into a career?
Yeah, like coach. Really, you work as a coach as well, like a career coach.
I guess it's part of our job as a mentor. It's really part of that, and I was lucky that indeed my postdoc mentor was very supportive, and we were talking literally from the first month there. We were talking about my future job, where it would be, what I would like to do, and it really helped me.
Yes, and now you're also doing that for your students now.
I hope it helps them, and when I talk to them, I feel they're all happy in their careers. So, I mean, I'm still a very junior PI. Like at 6th year, I started the lab, so most of them have like 1 or 2 years in their new position. So, I guess we need to wait some more years to see how happy they are and how good these choices were, but
you never know. But you can always switch again. And
exactly. I think but it's also the skill of thinking of your future and making decision, like long-term decision, is like a little bit what I was describing for my PhD. You know, I knew I didn't want to study this for my entire life, but this step was important for my future. So sometimes you can also get a job or a position to learn something specific to afterwards, do something that is closer to your interest. Or
Did you really make, like from the beginning on, did you make an outline for yourself, or did it just happen along the way?
Well, I guess I kind of had like a blurry vision, I would say. I didn't know I would become a PI, like leading a lab. I knew I wanted it, but I had no idea it was possible. I have to say that as I studied, there were not that many women in these type of roles. So, all the PIs around me were males. So most of the researchers were males, like technicians, and some engineers were women. And a lot of PhD students and some postdocs were women. But there was really this glass ceiling that was strong, and it did impact me, but I still thought I wanted to try.
Yes.
And I feel like there is a big evolution. So in the US, I think they're even more advanced, but also in France, to promote diversity in science.
Yes. Yeah. You're also a member of the ALBA network.
Yes.
Who, of course, promotes diversity in neuroscience. Can you maybe tell a little bit about this organization?
Well, so it's a really wonderful network that was spearheaded by Carmen Sandy, who is a former FENS president-elect. And actually, when she was leading FENS, she really felt the neuroscientific community needed actions to promote diversity, so women in neuroscience, but not only, diversity in general, and inclusion and equity. So she decided to launch this network, and I was part of it from the very beginning. I was still a postdoc or just very, very junior PI, so I didn't want to take leadership responsibilities, and I wanted to focus on my science, but I was an ambassador from the very beginning, so presenting the network at conferences and doing specific actions. So there are webinars that are organized very regularly to try to be more inclusive of countries that don't necessarily are able to come to conferences. And there are also just roundtables for people to discuss issues and the different problematics that you can have depending on the country or the continent you're working in. There's plenty of actions. There is a huge database on the website from publication reporting on biases and really giving the numbers, like where are we today, how is it evolving. And well, the good news, it's positively evolving. There is more women. The glass ceiling is still there, but it's at higher levels. So it's going in the right direction. There is also podcasts from researchers and young researchers, especially. There are interviews. So I did one of these interviews in the very beginning. And I think there are like 16 interviews of researchers, mainly women. And yeah, it's like more than 10,000 views. So it's, the network is starting to be very impactful. And there is plenty of action. So I would encourage everyone to become a member. You can just show your support to this cause. And then if you want to be involved, there is plenty of actions to contribute to.
Yeah, sounds really, really cool. And so they create a lot of awareness. What do you think is an important next step for how to improve the diversity and increase women like in higher positions? And
well, this is a very complicated question. And as I just said, I think it's very different depending on the countries. So, I could talk about my department in the university, but I think the first step is really raising awareness. And there are countries where it's much worse or it's much more complicated, where all girls don't have access to education. And so it's very dependent on the regions. Yes. So I think the first step, and it's what has helped really, I guess, in Europe and in the US, is to just point to the problem. So the first thing is really getting the numbers, tracking the representation of the people and the women in science, and try to have people be conscious of these biases and also raise awareness to the fact that having a diverse group is a more productive group. And that this has been shown in sociological studies many times, and that is really key, because some countries will just say, like, what's the point? Why would we include diversity? And it really helps to be more productive and to have a better balanced environment and richer environment.
Yeah, that's a really important message.
Yeah.
And also especially for the people hiring.
I could give you a list of like specific actions, but I think the actions are really dependent on the environment.
Yeah, talking again about your own career in academia, what challenges and obstacles have you faced during your career?
Well, I would say many. Many are inherent to science itself. Like, it is a challenging job to formulate hypotheses, and first like reading all this literature, formulating these hypotheses, and then designing the experiment to test them. Like, this is a very long process. I think the first challenge I had to learn, it was actually during my PhD, is like patience. Like, being patient is really key, and being excited and enthusiastic, I feel like it's easier, but really being patient and seeing things at the long term, I think, is a very important skill. And it was challenging for me. I'm not a very patient person. Let's say I wasn't. I guess I am much more now.
You learned it.
It's really an important skill. And doing a PhD, I think one of the most important things that a PhD will teach you is patience. Yes. And maybe a little bit of resilience, but which probably goes together.
Yeah.
And then I think today, as a PI, the biggest challenge is bureaucratic. It's like all the administration, which is becoming heavier and heavier, all the rules for everything. And it is a real burden on the scientists. 50 years ago, researchers in France, they would have like their own funding every year from the government. And it was small, but every researcher had some money to do research. Today, the system is very different, where you need to go get money. Experiments are much more expensive. They're like more comprehensive, including big data sets. And then you need the tools to analyze all that. So these type of science costs more money. So you need to go get these grants. I think this is challenging. I always think I love writing the science part of the grants. This is like, this is really fun. I enjoy doing that.
Also.
But that's like half of the grant. The other half is just like filling forms for plenty of, like the host institution and all the regulations that you validate. And I don't think we should not have all these rules. I just feel it's very heavy on the scientists, and maybe we should actually build a system where part of this burden is relieved from the researchers. So
How do you personally cope with that?
Well, I'm lucky in our institute that we have help, but it's a person for the financial department that is helping the 11 PI. So, I mean, she's wonderful, but it's a lot of work. So we still have to do a lot of things on our own. I feel like, more support, more permanent staff like that can help with that. I think that's important. And the other aspect, which is less dependent on human, but more on society, which is a challenge for me today, and I think a lot of PIs that are working in preclinical models, is this difficulty to talk about research in animal research in general. It has this negative connotation, and the legislation is becoming heavier and heavier, and there is some politics that wants to completely remove animal research, and I think this is really dangerous. I mean, we definitely need that, and most of the therapies we're using today for psychiatric disorders or cancer or many big disorders are based on research in animal models, and we really need them. And I feel really I'm struggling when there are interviews or people that come in the lab, make pictures and videos. They're like, oh no, we can't film animals. I'm like, but we need to show that. So I think one way I will to help overcoming that is when I became a member of the Fence Cavalier Network, is to try to have a way to impact society and tell them this is very important. And
because somehow, I don't know, the idea came that animal research isn't necessary or something. How do you think that idea developed in society?
Well, I think some people that really want to protect animals for good reason, and I think there have been some abuses in the past, and the humans' consideration for animals has really evolved over the last century, and I think it's good, but animal research is still necessary. And it's also the imbalance between how animal research and animals for food are treated, right? I mean, this is really questionable. Why should we stop? I mean, if we should stop animal research, then we should stop eating animals, but then this is not on the table, right?
No, well, it's also on the table, but for a
very few
small
people.
Group,
but so, finding places to voice and interact with society, to share stories and example of how much animal research is important, I think really is necessary. And
how was it for you to work with animals in a lab for the first time? Or
well, it actually was a real philosophical challenge for me. I mean, I remember it was my first internship, and I was studying this polymorphism of pro-BDNF, and I had a lot of mice. Actually, my project was 83 mice from 7 different strains that were in a condition where they were stressed, and then I had 120 mice that were controlled in another animal facility. So altogether, it was almost 200 mice, for one project. It was the project of a PhD that I was helping to work with, but I really had to think, do we as humans have the right to, take the life of these animals to answer our questions?
Yeah, and so you also felt that.
I mean, I really felt that, and I don't say we should not think about it, and we should not try to reduce as much as possible the use of animal. I just think that we need to use them to understand how, the brain works, and
there's no replacement for it. No.
I mean, some people are saying, oh, we should make a human brain model in a computer. But, well, first we need to have data to build this model. And most of the data are collected from animals.
So. Maybe in the far future, who knows?
Well, I'm not really convinced that this is the way we're going to solve how the brain controls different functions and how it's altered in pathological conditions. And
we don't understand enough of the brain to
make this model. Although I think modeling is super important, and we can gain from that. I just think it's not going to be enough alone.
Yes, Currently, you are a principal investigator yourself. What do you enjoy about supervising students?
Well, to see them learn, and it's I think, again, the patience I was mentioning earlier really comes into play here, because very often it's like planting seeds or I love gardening too, so I like this metaphor. It's, you plant seeds and then everybody has his own rhythm and will integrate, these new information or advice at different rhythm and, to let people grow at their own pace. And but it's really rewarding to see them blossom, basically. Yeah, I've been really enjoying that over the years. And, there are challenges too, but to see them grow is the best reward.
Yes, I can imagine. And also that you are a great example for your students.
Well, I hope so. I'm trying. I'm doing my best.
Yeah. Who was an example for you when you were in the beginning of your career?
Well, I was mentioning during my PhD and first postdoc, it was very male-driven environments. Yeah. So I guess my first real example was Kay, my postdoc mentor at MIT, really. And she was like really a young woman PI at MIT. Although I have to say in the US, they are more advanced in this, at least gender equity. Like in the department at the Picauer Institute, there were half PIs were women. Sorry.
At that time.
Yeah, already in 2012. So, and just that, it really hit me. I was like, oh, wow, it's possible.
Yeah, that's also an example of how important it is to have
absolutely
like examples when you're younger. Yeah, maybe to end this podcast, I always ask this question to the people I talk with. What advice would you give to early career scientists?
A lot of them. I would plant a lot of seeds. I guess the number one advice is like to follow their feeling or their aspiration, what they want to, like to not be brought down by people like trying to convince them that they're not made for that or but it doesn't mean like being rigid, right? It's also good to take inputs but really keep this dream and follow your dream, basically. Yeah. I really think it's and sometimes maybe like one of the advice is that, you have this long-term dream and then keep the direction, but sometimes the path is not what you expected. And I think this is very important. And it's hard because when you really want this position or like studying this project and the environment doesn't let you go like the straight path you had in mind, it's sometimes really hard. But my advice would be to follow advice and be open. I think also follow your dream, but be open to the path you're going to take.
Yeah,
not
alternatives.
Exactly.
Yes,
and you might very well end up to the same objective, but with a totally different career path that you had expected. So this is a little bit what we with Sami, my colleague with whom I wrote the perspective in EGN, we try to convey that there are many ways to study a specific question.
Yeah, definitely. Well, thank you very much for your very inspiring story about your own career, about your research, and the great advice you have for young scientists. I really enjoyed it. So
thank you.
Thank you
My pleasure
very much.
too. Thank you.
Thanks for listening to this podcast from the European College of Neuropsychopharmacology. The views and opinions expressed in this podcast are those of the interviewee and not necessarily the official position of the ECNP. And of course, this should not be the basis for clinical treatment decisions.